Good afternoon, everyone, and welcome to our presentation at EIA 2018. My name is Dale Usner, and I am the Chief Scientific Officer and Senior Vice President of Strategic Scientific Consulting at SDC. First, I'm going to share a bit about SDC as we give our online attendees time to log on. Then I will go over some housekeeping items before introducing Garrick for today's presentation. Thank you for joining the presentation and giving us a chance to demonstrate what SDC is all about. SDC is a specialized data service and strategic scientific consulting CRO. Operationally, we focus on biostatistics, data management, electronic data capture, interactive response technology, and data monitoring committee services. All things data in clinical trials. Earlier this year, we announced the official formation of our Strategic Scientific Consulting Division, focusing on clinical trial design and strategic decision-making, including regulatory pathways and due diligence for new product acquisition, with the goal of helping our clients optimize their clinical programs and portfolios. I am the head of this division, so if you have further questions, please feel free to see me after the presentation. Now for the housekeeping items. Before we get started, I want to make sure that everyone on the phone is able to hear me as well as able to see the presentation. As we have both internal as well as an online audience today, at the question section of the presentation, we are going to first address those questions that are on site and then follow up with the questions from online. As a note, we are recording this presentation, and later today or later this week, we will send out a link to this recording to allow people to review it online. As a note, this whole presentation will take 1 hour, including my time, including Garrick's presentation, as well as the Q&A. I would now like to take the time to introduce Garrick Wallstrom with his presentation, Optimizing Your Clinical Development Strategy by Identifying Promising Subgroups for Patient Enrichment. Garrick is a principal research biostatistician at SDC. He has over 18 years experience in clinical trials, bioinformatics, and public health disease surveillance. Prior to joining SDC, Garrick was an assistant professor at Arizona State University, where he was a top researcher. Where he was a top researcher in the College of Health Solutions. Garrick has been the principal investigator on 3 federally funded research projects, collaborated as co-investigators on numerous other grants, and served as an advisor to Hong Kong on public health preparedness during the SARS outbreak. Throughout his career as a statistician, researcher, and professor, Garrick has sought to innovate statistical solutions that yield insight into real-world challenges. He has more than 35 publications in peer-reviewed journals and is the co-inventor on 3 U.S. patent applications, as well as an additional awarded patent. Garrick earned his PhD in statistics from the University of Minnesota and received the Savage Award for his dissertation in Bayesian theory and methods. Without further ado, please join me in welcoming Garrick Wallstrom for his presentation on optimizing your clinical development strategy by identifying promising subgroups for patient enrichment. Thank you.
Hi, okay, so welcome again. I am here, I'm gonna repeat just for continuity here. I'm talking about a situation that we commonly face in clinical trials. Namely, we get to the end of a clinical trial, we have some tantalizing results, but we don't necessarily meet our primary endpoint. Very next question is, well, okay, we could pack up our bags and go home, but instead we want to try to get as much as we can out of that data and try to learn how can we better design the next trial. Let me consider an example. So this is a knee osteoarthritis trial. I'm considering this to be a early phase trial. The primary endpoint is a change from baseline in a weight-bearing pain scale. We're going to be comparing an active and a placebo group using a two-sample t-test. Because it is an early phase trial, we're using an alpha of 0.1. When we run the trial, we get our primary results. They're represented here in this table. So we have 60 subjects in both the active and the placebo groups. The mean for the active group is minus 0.4, so that is a decrease in weight-bearing pain. In the placebo group, we also have a decrease. So we have a minus 0.2 is the mean. So it's not as much of a decrease as we see in the active group. And when we compare these two groups using a t-test, we get a p-value of 0.1192. So we're not below our target of 0.1, but this certainly looks encouraging. So one thing we might do at this point would be to say, all right, maybe in this broader patient population, we don't have statistically significant efficacy, but maybe there's a subset of patients in which we see greater efficacy. So we could go back and say, all right, well, maybe if we just focused on patients that have a knee stiffness of at most 3, we could look just at that subset of patients and again, run the stats, compare those two groups, compare the active and the placebo. We now have so there's 52 and 53 patients in those two treatment groups. The mean decrease in the knee stiffness is now 0.5 versus 0.2 in the placebo. And when we compare these, we get now a p-value of 0.0133. Okay, that sounds better. We didn't lose that many patients, and now we have something that's significant. So this looks interesting. But of course, we could take this further. What would happen if we instead went and changed our criteria and looked at knee stiffness at most 2? So now we've got a much smaller population. We've got about half our original population here. The decrease in the active group is now 0.8. Okay, so a much stronger effect in this subset. And the p-value comparing these two, comparing the active and the placebo groups within this subset is now 0.0006. Ooh, that's something really good, right? This is really encouraging. Of course, this is only a couple of subgroups that we've looked at. If 3 was good and 2 is better, maybe 1 will be earth-shaking, or maybe not. So once we get down to 1, we now have really cut our population quite a bit. We've only got 15 and 11 subjects, and while the effects are roughly the same as we saw previously, the p-value is now 0.0660. So we've sort of run out of the ability to improve this subgroup. But of course, here I've really only been thinking about baseline knee stiffness for trying to identify an interesting subgroup. But there could be other ways in which we could identify or create subgroups. Okay, so we know that this subgroup with knee stiffness at most 2 looked really promising, but we could explore more. So maybe we want to do something where we combine both knee stiffness and another characteristic, such as BMI. So maybe we could look at subjects that have knee stiffness at most 2 and BMI at least 24, go through the same process, calculate a p-value, we get something of 0.0015, so that actually still looks pretty decent. Or maybe use BMI less than or equal to 30, that too looks pretty good. And we could obviously keep repeating this process. So this is generally the topic that I want to discuss today. How do we find high efficacy subgroups? How do we find a subset of patients in which we see greater efficacy than in the broader patient population. And why do we want to do this? Well, we primarily want to do this because it guides us towards making better decisions in future trials. It allows us to do patient enrichment. It allows us to suggest strata in future trials, and maybe it'll even allow us, if we're thinking in terms of publications, if there's a particular subset of patients that we might want to focus our analysis on. But I'm primarily thinking in terms of the impact on a future trial. So I'm going to discuss, in contrast, really two types of approaches that are used to define these, or identify these subgroups. One being the human-driven approach, and the second being a data-driven. Thank you. The human-driven is really the kind of logic that we just went through in this knee osteoarthritis example. Namely, we can identify a subgroup using clinical knowledge, using expertise, go and run the statistics and test things, and based on those results, maybe refine what that subgroup looks like, what the criteria are, again, calculate the results, review the results, and refine again. And we could go through this sort of iterative process. It can be a little slow because there's a lot of manual intervention going on in there, trying to review the results and reconstruct a better subgroup to examine. But it is what it is. So this is a pretty common practice. And the question comes, okay, so at the end of the day, we find a subgroup. How do we know it's real? How do we know it's going to reproduce? Of course, the real answer is we don't. We never do. It's statistics. But what we'd like to do is we'd like to have some assurance that it's not likely to be due to chance alone. Okay? That it's not just a spurious finding that was found because we tested lots of possible subgroups. So I need to bring us back to your beloved Stat 101 course and just mention type 1 error for a moment. So type 1 error rate in general is the probability or the likelihood of getting a significant finding by chance alone. In this context, it's the likelihood of finding a subgroup by chance alone. And it's really important that this is controlled. Okay? Why do we want this to be controlled? Well, if this type 1 error rate is really high, then that means you've got a high chance of finding an interesting subgroup by chance alone. And if you find an interesting subgroup and then go on to spend millions of dollars exploring that subgroup in a future study, that's not probably a good use of money. So we really want this controlled so that whatever subgroup we do find, we have a reasonable assurance that it is not due to chance alone and that it's something real and worth pursuing. There are a couple of causes of type 1 error rate inflation, and I'm really going to focus on the first one, which is multiple testing. Every time we test these subgroups, or we refine and test subgroups, we're doing more statistical testing. And as we look harder and harder and harder to try to find an interesting subgroup, our chance of finding something by chance alone increases. So this multiple testing really can drive up the type 1 error rate. There's also an additional cause of inflation, which is really just something that always comes along with doing post hoc analysis. That's why we differentiate post hoc analysis versus planned analysis. As soon as we're doing post hoc analysis, what kinds of, in this context, what kinds of subgroups we examine may be based on the results of the trial, and hence there's sort of this implicit but hard to measure inflation of the type 1 error rate. Okay, so pros and cons. One of the big pro here is that the subgroups that we examine in the manual approach are practical, right? They're being constructed using clinical knowledge, and they're thought about in terms of this might be something that we want to use in a future trial for an inclusion-exclusion criteria. So they're very practical groups. On the other hand, it's a manual and very slow process where subgroups have to get defined, tested, reviewed, refine the subgroup, test it, review the results, etc. That can take time. May take many months to go through that process until people decide to stop. And importantly, there's no type 1 error control here. So there's multiple testing, might be testing half a dozen or a dozen subgroups, and by itself, there's no control for that multiple testing. So the data-driven approach is, in theory, very similar to the manual approach, except the process of selecting which subgroups to examine is now not being done using clinical knowledge, it's being done by machine. And so in this process, the data will drive which subgroups to look at. They can then be iteratively tested and searched over, and it can be used to identify what's the best subgroup out of some collection of possible subgroups. The one big downside to this approach is that the subgroups by themselves may not be very practical. And this really sort of depends a bit on how this is set up. But typically it's not being, there's no sort of clinical input in terms of what subgroups are examined. It's more of just sort of a data mining exercise. So the subgroups that might come out might look like something like BMI divided by knee stiffness less than 8.2. Oh, that's a great subset of patients to look at, which is fine, and it might be true in a certain sense, but it might be very awkward to use as an inclusion criteria. The benefit to the data-driven approach is that lots of possible subgroups could be examined, right? So if we are able to very quickly look at thousands of possible subgroups, test them, and find the best, hey, now we've done a pretty good search over that space of what subgroup might be interesting. Much more thorough than looking at maybe the best 6 that come to mind. The type 1 error rate here is also not controlled, and it's probably even worse than what we see in the manual case, because now we may be doing extensive multiple testing. We may be testing not just a dozen or a half dozen subgroups like we saw in the manual case, but instead we might be testing thousands or tens of thousands of subgroups. There's a lot of multiple testing that could be done here. All right, so I want to talk about a better way, okay? And this is an approach that we developed at SDC. It's called constrained search and permutation evaluation. And it really has 2 parts to it. One is that we want to be able to automate the search, so take some of the ideas from the data mining type approach, automate the search, but at the same time, we want to ensure that the subgroups that we find are reasonable and practical, okay? That is, we want to impose human intelligence into the process. At the same time, we think it's really important to actually control that type 1 error rate, be able to assess how likely it is to be due to chance alone. So the constrained search part of this really means defining what kinds of subgroups we actually want to examine. There's lots of possible subgroups that are probably not going to be sensible, and we don't want to waste time looking at them. In this context, one of the first things that we want to consider is what are the baseline characteristics that we would want to use to define our subset, okay? In the osteoarthritis example, it might be a baseline BMI, baseline knee stiffness scale, baseline pain scale, baseline physical function scale, some basically baseline disease characteristics. We also want to think about, we might use these characteristics by themselves, but we also might want to combine them in some way. And so we want to ensure that combinations are going to make sense. So something like what we saw previously, baseline knee stiffness of at most 2 and baseline BMI at least 24, that may make sense as a combination. But doing something crazy like BMI divided by knee stiffness, not so compelling. For many of these, there might be cutoffs. So for something like BMI, it would be reasonable to limit the cutoffs to the integers. While you could have a cutoff of whatever, 24.3, that's might not be something that you really want to think about seriously. The number of characteristics that are used is also something that you'd want to think about. Basically, you don't want necessarily an overly complex definition of a subgroup. Maybe we have four different characteristics here, but we probably don't want our definition of subgroup to use all four of them. We want it to be fairly simple. So by limiting the number of characteristics that are used, that allows us to limit the complexity. And finally, one of the things that we want to be able to do is, so there's two parts to this, but limiting the number of subjects that are in the treatment groups is also a desirable or reasonable consideration. Okay, two reasons for that. One is, of course, we want to have enough subjects so we can actually properly evaluate and compare the active and the placebo group in using this existing data. And going forward to a future trial, if this number is too small, then that means that future enrollment is also likely to be very difficult if we're looking at too small of a proportion of the total population. The second part to this is the permutation evaluation. And this is probably the most technical part of this talk, so bear with me. What we have on the top is what we do with our original data. So we've got our original data. We apply a search algorithm in order to identify the best subgroup, called subgroup star. Okay, we then apply whatever our test is for our primary endpoint and calculate p-value star. We then can repeat this process on permuted data sets. So each one of these permuted data sets is just like the original data, but where we randomly scatter the treatment groups that patients are assigned to. Okay, so we're just permuting the labels, this is how we often speak of it. But the rest of their data remains the same. So we can scatter their labels, scatter their treatment assignments, and then repeat the process of searching for the best subgroup and calculating a p-value. And we can do this over and over again. What's nice here is that at the end, these M p-values represent p-values for what would we expect to find if there was in fact no difference between the active and the placebo groups. Okay. So this gives us sort of the baseline to which we can compare the p-value that we found with our real data, the p-value star. If the p-value star looks just like one of these guys from the p-value 1 to m, then our subgroup is probably not very outstanding. On the other hand, if it's much lower than the typical p-values that we see, then that suggests that maybe indeed we do have an interesting finding. So this approach really meets all four of these properties that we've talked about. The subgroups are practical, and they're practical by design. We force them to be that way, right? Because we think about it carefully in advance, what kinds of subgroups we want. It's automated, and we can still examine a large number of subgroups. Even with these constraints, there still may be thousands of possible subgroups that we want to be able to search over. That's still not something we want to be doing manually. And importantly here, the type 1 error rate is something that's controlled. So even though we're doing searching over hundreds or thousands of possible subgroups, we're controlling that type 1 error rate. If I come back to the knee osteoarthritis example, we initially here started with 3 baseline characteristics and said, all right, let's use at most 2 of them. I also want to require that there are at least 10 subjects in each treatment group. When we applied this method, the best subset was one that had baseline knee stiffness of at most 2 and baseline BMI of at most 32. Okay, so it's very similar to the initial one that we found just sort of in the manual search of knee stiffness at most 2. You might recall that p-value there was 0.0006. The p-value for the subgroup here is 0.0003, so it's slightly better. But of course, it's also a slightly smaller population. So here we've got a minus 0.8 as a mean effect in the active group and a minus 0.1 in the placebo group. And it's highly significant by the t-test. However, when we evaluate this using CSPE and do the permutation test to accurately assess what the true type 1 error rate is, we see that it is still significant. So it's still a very interesting subgroup to explore. But I probably don't get quite as excited about a 0.03 as I do a 0.0003, at least as a statistician who gets excited by p-values. So this allows us to really temper what the expectations are. The expectations for seeing a p-value of 0.0003 might be, oh, this looks really outstanding. A 0.03, I think, creates a little bit of a different response. Okay, so I want to talk a little bit about evaluation. So we have this technique. Let's look at how it works. And I'm going to talk about 2 different scenarios. One in which we use simulated data. The use of simulated data is nice because we can do things like actually measure error rates. How well does it do at finding true high-efficacy groups, and how well does it do at not finding groups when there are none? We also are going to look at real study data and just to really, in this case, get a qualitative assessment of how the technique works. So for the simulation, there are really 2 scenarios, as I said. In scenario 1, there's no true high-efficacy subgroup. So we'll use this to say, all right, well, I can run the technique here. Anything I find, if I find any subgroups, they're false positives. I don't really want them. And that allows me to assess the type 1 error rate. For scenario 2, we do the simulation in such a way that there is a high-efficacy subgroup by design, in this case, containing about 60% of the subjects. We can then see how often do we find this subgroup and use that to measure type 2 error rate or power. So we'll be looking at the type 1 error rate, the type 2 error rate, and we'll be comparing these between the CSPE and what we would view these if we just did the typical manual search for subgroups. And so here are the results. On the left, we have the traditional search. The big blue bar is the type 1 error rate for the traditional search. It's at 78%. Okay, 78%, right? That's pretty big. What does that mean? That means that, okay, well, first of all, this was not a huge search, right? We only had 3 baseline characteristics that we were looking at. When there's no differences between the active and the placebo group, we have a 78% chance of finding something significant anyway, right? Which means if your next action is to look at that subgroup and start a new trial focusing in on that subgroup, that's a 78% chance of doing a trial and wasting that money pursuing a group that is just based on chance alone. If we consider CSPE, the type 1 error rate was 10%. Here we limited it. We were targeting an alpha of 0.1, so we were right on track. And the type 2 error rate was 9%. That means 91% power. So we had a 91% chance of finding a subgroup when in fact there was a real one. Okay, so that's a reasonable trade-off for controlling that type 1 error rate. Okay, so let me look at real study data. So for confidentiality reasons, I can't really say very much about this trial, but this is a phase 3 trial. It had more than 300 subjects. There were active and placebo arms in this trial, and the primary endpoint was interesting in that 0.05 to 0.1 range. So it's interesting, but not below the 0.05 target. In this case, there were more than 40 possible disease characteristics that could be considered. So you can imagine if you started doing this in a manual approach, looking and trying to form subgroups based on more than 40 characteristics, even doing them one at a time is painful. And certainly once you start talking about combinations, very, very painful. So when we applied CSPE to this, one of the first things was to identify, well, some of these baseline characteristics, maybe they're not as interesting as others. So we narrowed it down to a set of 15 characteristics, which is still a pretty large number, and maybe ideally would have been a little bit less than that even. But sometimes it's hard to throw out baseline characteristics that you think, well, maybe that's the one. So here we limited it down to 15. And we wanted to ensure that we want to use at most 3 out of those 15 in our definition of the subgroup. From there, we used sort of reasonable cutoffs for these characteristics, and we required there to be at least 50 subjects in each treatment group. Okay, again, that's mostly towards the idea that we want enrollment to not be a complete nightmare in a future trial. In this case, many of the subgroups that we found had p-values that were small. There were many subgroups that gave p-values that were small. The best subgroup that we found used 3 characteristics and contained about 40% of the population. Its initial p-value was 0.005. Okay, so it's pretty significant. However, when we applied this technique to it, the p-value was 0.25. Okay, so even though the original p-value here was highly significant, the p-value using the permutation test was not, suggesting that there's certainly a reasonable chance that this would be due to chance alone, and may not be as, maybe worth further examination, let me put it that way. Okay. So to summarize, we developed this approach, CSPE at SDC, and it really gets all 4 of these points. Sort of by design, it allows us to focus on practical subgroups. It's automated. We can still search lots of possible subgroups. And very importantly, we can control that type 1 error rate so that we have some assurance or some idea of whether this is likely to reproduce in a future trial. I want to say a few minutes about practical considerations. Generally, the hardest part here is really identifying, sort of two worlds. It's usually easy to identify what are the baseline disease characteristics that might be of interest. The only challenge is if there's a real lot of them, like in the example where there are more than 40, paring them down to something and to ones that you really care about can be a bit of a challenge. But after the characteristics are identified, pretty much the rest of the constraints take care of themselves just by posing normal human logic to it. The variables themselves will naturally impose constraints. One of the things that we did think about is how complex of a subgroup do you want, right? So we saw before we did things like limit it so that it would use at most 3 of those baseline characteristics. But one could also do things like find the best subgroup that uses at most 1, find the best subgroup that uses at most 2, and at most 3, and calculate all of them and get p-values for all of them. And then those can be compared. And that's really more broad than that. There's no reason, so everything I talked about was really talking about finding one subgroup, the optimal subgroup. but there's no reason you have to stop at one. You could use the same technique to identify maybe a half dozen of the best subgroups. Once you find these subgroups, you can then consider them, bring in some clinical judgment, consider what the impact will be on enrollment, and then choose a subgroup from that point. So there's really two different spots in the clinical trial process where this could come into play. The first is in the actual design of the clinical trial. One could specify in advance what kinds of subgroups you're going to examine. In some ways, this might feel a little bit odd because you're designing a trial and you're planning on an analysis that's only going to be useful if it fails, maybe, or is most useful if it fails. But it's useful to think that way and be prepared. So that's one spot, is actually in the design of the trial. The second spot where it comes up most naturally is in post hoc analysis. So after the trial is over, the results aren't quite what you want, then you want to see, is there a better subgroup? In both cases, we can use the technique to identify interesting subgroups, to facilitate patient enrichment in future trials, or support stratification in future trials. Okay, so to summarize, so we developed this CSPE approach for identifying high-efficacy subgroups. It, by design, finds practical subgroups that can be used, and it controls the type 1 error rate. Okay? Again, that's really important here because if we don't control that type 1 error rate, we're likely to find things that will not have reasonable chances of reproducing in future trials. The main value of this is, what does it mean about the design of future trials? And by identifying subsets of patients where we see greater efficacy, we can then focus the future trials on those subsets of patients, move them into inclusion-exclusion criteria, or use them to define strata and improve the quality of that next trial.
Thank you very much, Garrick. I'd like to take the time now to open up to questions and answers.
Thank you. It's very interesting presentation. One comment I want to make is that what is probably missing in the literature review. I mean, there were tons of papers and methods that emerged in the last 7 years. Actually, I wrote a paper together with some colleagues, published in medicine, called the tutorial on data-driven subgroup analysis, not to promote myself, but this tutorial has 100 references. Not all of them are nice, but maybe 50 of them are. And in particular, one method that we developed called size, kind of similar to this, also has to strain search with permutation based population error. So I think it's important to look at the landscape of different methods available. Now you can compare everything to a traditional approach, which is completely processed. But it's not that everybody uses to enforce a lot of different methods, but also combination of machine learning, causal inference, and multiple testing. And so, of course, people who would be thinking about what method to use, we need to know the landscape. Thanks.
Any other questions? Are there questions online?
I do have a question from online. We have a webinar study asking, why do you scatter the labels in permutation evaluation?
So the question was, what is the utility of scattering the labels in the permutation evaluation?
So I'm trying to get that there. So the idea behind the permutation test is that by scattering the labels, we're making our data as if the treatment assignments were random. That is, there's no association between the patient's data and their treatment group. So it gives us it's really considering the situation where our null hypothesis here, if I get too statty, the null hypothesis here is that there's no difference between the active and the placebo group. And if that's true, then switching the labels around doesn't make a difference. And so this gives us a way to assess what the how likely it is to have the p-value that we found occur by chance alone if there are in fact differences between the treatment groups.
Thank you, Garrick. Are there any further questions online? Okay. If not, I would like to thank everybody for joining us today for this presentation. Thank you very much, Garrick, for the wonderful presentation. And we still have food and beverage outside, should folks want to partake on that. Have a great rest of your day in conference.